Okay, so let's talk about what we've learned so far about DNA, RNA, and then we'll continue talking about transcription. So what we know so far is that DNA is a double helix molecule. That means it's double-stranded. It's made up of sugar and phosphate backbones, which are connected to nitrogen bases. There are 4 different nitrogen bases in DNA: A, T, C, and G. These 4 nitrogen bases make up the steps of the DNA ladder. They're connected by hydrogen bonds. The sugars and the phosphates, which are the sides of the ladder, that would be these red lines, those are connected by what we call covalent bonds. All right, remember DNA contains our genetic information in the form of genetic code. The genetic code, if you remember, is A, T, C, and G, those letters. The genetic code contains instructions for making proteins, but the problem is that DNA and proteins don't speak the same language. So they can't talk to each other. So DNA has the instructions, and proteins are what we need to express our genes, but they can't communicate because they don't speak the same language. So remember, we have to use an interpreter called RNA, because RNA can talk to DNA and it can talk to protein. So it interprets both languages. It's a translator. There are 3 types of RNA. I don't know if you recall them from earlier this week, but they're mRNA, tRNA, and rRNA. Each of these types of RNA has a different job that it does. Okay? Before we continue with RNA, we've learned also something about DNA. We learned that DNA makes a copy of itself during a process called replication. Okay? We learned that mRNA, or we started to learn yesterday a little bit about mRNA and how it's made during a process called transcription. During transcription, we're going to take DNA and we're going to make it into mRNA. So we're going to use DNA as a template to make a strand of mRNA. And remember, we need that RNA because RNA is going to be our translator between DNA and proteins. Remember, amino acids are the building blocks of proteins. So eventually, we're going to take this RNA that we made and we're going to make proteins out of it, or amino acids. That process, which we're going to learn about a little later, probably tomorrow, is called translation. Okay? Okay, so we've reviewed what we've talked about so far in the last 2 weeks. DNA structure, DNA replicates itself, and we know we need RNA to be the translator between DNA and protein. So now we're going to talk about the process of protein synthesis. Remember, the word synthesis means to make something. So in this case, we're making proteins. The process of making proteins is called transcription. This is the point where you need to begin writing on your handout. You should already have that out, but just in case you don't, you want to get it quickly. We're on the 2nd page of your handout where it says transcription. Some of you began writing on this page yesterday. Some of you have not started writing about transcription yet. So I'm going to start as if we're starting from the very beginning. So whatever parts you've already written down, you don't have to write them again. So here we go. Okay, so we're not going to watch this video right now, but this video is on your handout. So the web address is listed on your handout. So if you want to watch it later to better understand the process of transcription, you can do that on your own. Okay, so let's get to talking about transcription. Some of you already wrote this down yesterday, so you don't need to write it again if you've already written it. If this is your first time seeing this, you will only need to write the highlighted part on this slide. So you'll notice a picture of a cell over here. That picture of a cell is there because I'm trying to remind you where transcription happens. Transcription happens in the nucleus of our cells, right in there. And transcription is the process, remember, of making RNA from our DNA. We use the genetic code in DNA, the AGCT bases, that's the genetic code. We use that as a template to make our mRNA molecule. So we're going to use DNA to make RNA. The sequence of bases in the DNA allows us to know how to make our RNA. So we're going to match up complementary bases in the DNA into a new strand of RNA, and I'll show you how that's done in a little bit. Remember that RNA is a translator between DNA and protein. So later on, that RNA is going to also be used to help us make amino acids. And amino acids, if you remember, are the building blocks of proteins. Remember, there's also another process we talked about a while back, actually about a week ago. And that process is called replication. That's where DNA copies itself. That also happens over here inside the nucleus. Okay, now let's get into the steps of transcription. Transcription occurs in 4 major steps that I want you to know. The first step is that DNA helicase unwinds DNA into 2 separate strands by breaking the hydrogen bonds. Remember, those connect the bases, okay? Just like helicase does in replication. So remember, helicase sounds like helicopter, and it unwinds the DNA. It's doing the same thing here. Because remember, we're going to use the DNA to make our RNA. Also, all of these steps you need to write down. So everything on this page over here, you need to write. The second step is that now that the DNA is unwound, we're going to match up our complementary RNA bases with it. So we're going to make a single strand of RNA using our DNA that we split apart. Remember, in DNA, our bases are A, T, C, and G. And remember, in RNA, our bases are C, G, A, and U. There are no T's in RNA, none. If you see a T in the RNA strand, there's a mistake. Okay? So, let's talk about our base pairing. C's are going to still match up with G's, just like before. T's are going to match up with A's, just like before. But If you see an A in the DNA strand, it's going to become a U now in the RNA strand. Because remember, there's no T's in RNA. We'll show an example later, so you'll catch on quickly. Make sure you wrote all the steps so far. All right, the last two steps of transcription are really important. So, step 3, after all the pairing is complete, a single strand of RNA has been produced. So once we match up our C's and G's, our A's with T's, and our, oops, I made a mistake, A's don't match with T's, what do they match with? That's right, U's. So after we get done matching up our C's and G's, our A's and U's, and our T's with A's, then we have a strand of RNA that we can use later on to make proteins. Step 4, RNA and DNA are read in a sequence of 3 bases, or 3 letters at a time. So no longer reading just single letters, A, G, and A. We're going to read AGA together in a triplet like that. This triplet of bases is called a codon. So from now on, whenever you write DNA or you write out RNA on your paper, you're going to write it in 3s. So you're going to write the bases 3 at a time. This is called a codon. And codons are really, really important because they signal which amino acids we need to make later on. So they tell us which proteins we need to make later. And remember, we need those proteins to express our genes, our brown hair, our blue eyes, etc. At the bottom of the page, you'll see examples of codons. Make sure you write this. So you should have written all of this on your paper, okay? So at the top, in the black letters, we have codons that represent a sequence of DNA. In the bottom, we have in pink codons that come from a complementary RNA strand that we've just made. Notice the pairing. A's match up with T matches up with A. C matches up with G. A matches up with U. Okay? G's and C's. T goes with A. C's and G's. T's and A's. G and C. A and U. CG. T goes with A. So remember how they pair up when you're making your strand of RNA. There's no T's down in that pink section. Okay, so we've talked about the steps of transcription. We should know what a codon is. Okay, and now we're going to talk about how to actually do a transcription as practice. So we're going to transcribe this DNA into a strand of mRNA. Okay, so remember now we have to read in 3s. So we have to read our bases in codons. So I'm going to draw lines here to separate my codons out so I can see them. We have a little extra base there at the end, an extra C at the end. That's okay. Sometimes your DNA is not evenly numbered. So if I want to make RNA from this DNA, the first letter I should write underneath this A over here is what? That's right, a U. And then the next letter should be what? C. And then C again. And if I keep going, I should have an A here, a U here, a G here, and then C, A, and then a G. Okay, we want to make sure that we still write in codons and triplets. Okay, so to help you keep it straight, you can put the lines there so you can see the codons. All right, so now you've practiced. Now you need to go to your worksheet and answer all the questions on the last 2 pages about transcription and RNA. Once you're finished, you need to show this to Miss Johnson so that she can make sure it's done. All right, have a good rest of the day.